final presentation in this session. It's from our own lab. So proudly present Sina, so a senior PhD student in my lab, is almost coming towards the end of his PhD, and he's done also a lot of work in the modeling, especially multi-scale models, and I think that's really great how the community is now sharing everything so rapidly and freely, so you can really build quickly and extend work from other groups. And so Sina's going to present some of our own work in that research field.
Hello everyone, I'm going to present on future of multi-scale modeling in NIBS today. My name is Sina Shirinpour. I'm a PhD candidate in the OPITS lab at the University of Minnesota. So first, let's go behind the rationale behind multi-scale modeling, why we do it. Just a quick introduction. So far, you've learned a lot about the importance of electric field modeling in non-invasive brain stimulation. But one thing that is missing is that there is no direct, straightforward physiological response between electric field and neuron activity. And even those relationships are typically non-linear. So, and because brain is pretty complicated, we still don't have a clear understanding of the mechanism of NIBS, and especially because in vivo measurements in humans are pretty challenging. Modeling is one of the tools that can help us greatly to understand what is going on at the neuron level when we apply NIBS. So modeling have been used extensively in the previous studies, but typically they, like for this particular question, they either do a macro scale electric field modeling or a micro scale neuron level modeling. And each of them have their own pros and cons. For example, at the electric field modeling, we calculate electric field at the whole brain level, and we typically deal with the physical effects of the NIBS, like calculating electric fields, and we have to take into account brain structures and electrical properties of the tissues in our model. But there is no direct physiological estimates of neuron activity. On the other hand, at the neuron level, we're working at the cellular level, and we can directly estimate the physiological response of the neurons to external electric fields. And we have to deal with like morphology and biophysical properties of the neurons and channels and everything. But typically in those studies, there is no realistic external electric field simulated. So the whole goal of multi-scale modeling is to combine these two domains of modeling. And throughout this presentation, we have used SIMNIBS for electric field modeling, and you're familiar, it uses FEM modeling to calculate electric field. And for neuron level modeling, we use Neuron package that uses cable equations to estimate the behavior of the neuron, like the electrical activity of the neuron. So now that we know why we should do multi-scale modeling, I'm gonna explain a little bit of what has been done previously. So generally speaking, the idea of multi-scale modeling is not very new, but most of the previous studies have used very simplistic forms of the multi-scale modeling that cannot capture the whole like complicated nature of brain. And because of the like huge computational restriction and limitation of the modeling softwares that we've had so far, it's been only in the past few years that we've seen more realistic multi-scale modeling. Here are two of the examples, like the Abera model from Dr. Peterchev's group, and in previous talks, they have referred to this a little bit, and I'm gonna talk more about this later. They have studied the effects of tMS on neurons, And another example is Seo and June, that they studied the effects of tDCS on neuron activity using multiscale modeling. And based on these recent literature, on top of the two that I've mentioned, it seems that this multi-scale modeling is a very promising new tool, and I would say it's kind of the future of modeling for NIBS. But we are still at the beginning of the road, and there needs to be a lot of more development in our modeling and many more studies to come to understand more about like the scientific questions that we want to answer. For example, one of the main issues in the multi-scale modeling fields is that previous studies typically have not included like synaptic input into the neurons, and basically the neurons are silent besides the external NIBS electric field that we have. But in reality, neurons actually receive a lot of synaptic inputs, and neurons are basically never silent. And this intrinsic activity of the neurons plays a major role in neurons' response to NIBS, and especially if you want to measure the subthreshold activity of like tACS, because tACS is not strong enough to directly activate neurons, but we know that it can modulate activity of the ongoing oscillations of the neurons. So in the rest of my presentation, here's some of our goals to extend previous models to include active synaptic input and like bridge the gap between human and monkey brain, because we there has been a lot of experimental studies in monkey brain, and multi-scale modeling is a great way to translate those what we know in the monkey brain to humans. And I'm going to talk a little bit about how to model plasticity in neurons as well. So with that background, I'm going to explain quickly how we do multi-scale modeling. So you've learned so far how we can do electric field modeling in SimNIBS. We mainly talked about humans, but we are not limited to that. We can talk about, like here we have the human brain, like a monkey brain. We can even use like an in vitro model, which has a like a brain culture, a gray matter culture put in a CSF matter. Like to simulate what happens experimentally. What we can use that in SimNIBS as well. With the FEM modeling, we can calculate all the electric fields induced in the brain or the tissue culture that we have. But after calculating those electric fields, it's important to couple those electric fields to the neuron scale, like the external potentials at the neuron level. And I'm not going to go through the details, but basically we calculate something called quasi-potentials from the electric fields. And we can calculate based on those electric fields the external potential due to NIBS at the neuron level. And here, for example, we see a gradient from one side to the other side of the neuron. And with that information, we can use a neuron package to simulate the behavior of the neuron under such external field. And that was the spatial distribution of the fields, but also it is important to take into account the waveform of the stimulation, for example, tMS or tACS or tDCS. By having the temporal and spatial distribution of such electric fields, we can put everything together and have a multi-scale modeling and see what happens at the neuron level activity. Here, because I don't have much time, I'm not gonna go through the details. I'm just gonna show some examples of what we can do with multi-scale modeling and how wide of the questions we can answer with such tool. For example, this is the first thing that when I started doing in multi-scale modeling was tACS entrainment. this is like the tACS electric field induced in the brain, and I used a very simple model at first, which is a ball and stick model that includes soma and dendrites. And we applied an tACS, like sinusoidal electric field, under a synaptic current injection that replicates the behavior of the neuron under synaptic inputs. And what we see here is that during the time that tACS is off, orange shows the stimulation, and the blue shows the behavior of the neuron, like spiking of the neuron. When the stimulation is off, we see that neuron randomly spikes due to the synaptic current injection. But after we turn on the stimulation, we see that the neuron spiking gets time locked with the peaks of the tACS. And basically, this is showing entrainment, which is like one of the strongest hypotheses of how tACS affects neurons. And with that promising results, we moved on to a more complicated model. And so, like in the previous studies, like there has been a by Abera, Peter, Chef, and Grill. They have shared these sophisticated neuron models that I would say are the state-of-the-art models. They use like very realistic morphologies from Blue Brain project, and they have very accurate biophysics of the neurons. Here, for this presentation, I'm going to show the results for only one of these neurons, which is the layer 5 pyramidal neurons, because these are one of our prime suspects for how NIBS is activating neurons. These seem to be the responsive neurons. But using multiscale, you can study all types of neuron. So we did the same principle that I used for tACS entrainment, but I used this sophisticated pyramidal cell. And here we can see the raster plot of the neurons, which basically simplifies the whole results into just spike timing. That shows when the spikes happen throughout time and trials. For example, at trial 1, we have a blue dot that shows there was one spike happening at like 10 milliseconds, and then there is another spike at 60 milliseconds. And now we have the spike timing over the whole trials. And we see when the stimulation is off, we have some random spiking, but after stimulation is on, we see more dense spiking activity during the peak of the stimulation and less activity during the trough of the stimulation. And if we average everything together, we'll see a histogram that nicely represents the entrainment, that we see more activity that is time locked to the peak of the tACS. And here is like the dependence of the entrainment to the intensity of the tACS. I'm not going to go through the details, but if we increase the amplitude as we expected, we see more entrainment. And then I'm going to change gear a little bit on talking about tMS and how tMS affects neuron. This is the same pyramidal neuron that we have. And when we apply tMS, a biphasic pulse, we see that there is a depolarization happening at an axon terminal, and action potential is propagated through the soma, and after that everywhere in the neuron. And so for this neuron, these are apical dendrites, and most of the other branches down here are axons. And it is important because there is still a controversy of how tMS activates neurons. Like some people say, like soma is the place that is activated, but using these accurate multiscale neurons, we can measure what models suggest neurons activate the site of the action potential. But it's important to note that thresholds of action potential, for example, here, if I apply 100 volt per meter action tMS, I see neuron firing, but if I go lower than that, a neuron will not fire. So with modeling, we can estimate threshold of the action potentials. But it's important to note that threshold depends on the neuron type and the E-field orientation and a bunch of other parameters. And for example, like in the Abera paper that previously stated, they have studied such electric field orientation, and this is basically a replication of like tMS threshold dependence on the neuron activity we see here. That basically in a spherical coordinates, if we apply electric field in different directions, we see that threshold depends on the exact orientation of the electric field. For example, in this region, we see the lowest threshold, and that corresponds to an electric field alongside the somatodendritic axis. And in the field, we already knew that somatodendritic axis, like from the apical dendrites towards the soma, we see the highest responsiveness to electric field due to tMS, but these results show that that is not the only orientation that tMS can activate neurons. There are some other regions depending on the neuron branching that can also activate neurons. So as we can see, these results suggest that just looking at the normal component of the electric field at the wall of the sulci will not explain everything that is happening. So multi-scale modeling can help us understand such things. But then we took the multi-scale modeling into another step, and it's looking at the subcellular dynamics of the neuron. Specifically, we looked at the calcium concentration of the neuron. And the reason is that calcium ions play an important role in a lot of dynamics of the neuron, specifically for plasticity. We're interested in studying calcium models. And with the help of some collaborators, we're doing some of these calcium concentration modelings, and there are a bunch of equations that we numerically solved after having the membrane potentials. And here's some quick results that we have. So basically, we have applied an rTMS to a neuron, a realistic neuron. And when I play the video, you'll see the calcium concentration throughout the neuron. And what we see is like a sophisticated pattern of propagation of calciums across all the branches in the neuron. And what we see is the average of the calcium concentration throughout the neuron. And we see at each tMS spike, we see like calcium goes up and down. But what is interesting is that these models suggest that, for example, if the tMS pulses are close enough together, calcium concentration doesn't go to baseline, and the baseline of the calcium increases over time. And with these results, we can understand more about what is happening at the neuron level and explain furthermore about the plasticity and long-term behavior of the neuron under NIBS. And I want to specify that I showed some of the capabilities of the multi-scale modeling and what answers we can give to some scientific questions. But I want to make sure you understand that these are not the only possible things that you can answer. There are a lot of many other questions that are important in the field and multi-scale modeling can help with. For example, when you apply NIBS, what type of neuron react? Under what circumstances neurons react? And what is the role of morphology and biophysics of the neuron in NIBS activation? How to translate the results between different species, for example, monkey, mouse, humans, and a lot of other questions. And With that, I'm happy to inform you that pretty soon we're going to release our open source multi-scale modeling toolbox with a bunch of instructions and all the codes necessary to be able to start doing multi-scale modeling in case you're interested. And this is just a very brief overview of the general procedure. We do modeling in 3 levels, like electric field, neuron scale, and subcellular calcium concentration. At the electric field, we basically use NIBS. You can use any head model that you want, like animals, in vitro models, humans, and calculate the electric field, and then couple the results to neuron. And in our code, we have made it available so that you can use any morphology files from neuron reconstructions and automatically generate the neuron models in the neuron package and apply, for example, a tMS train to simulate what happens at the neuron level under rTMS. And after getting the voltage membrane results from this level of simulation, we can use those results to calculate calcium concentration and answer some questions about plasticity and things like that. And I think with that general overview of the multi-scale modeling, I hope that you learned something and stay in tune for our toolbox. And with that, I want to acknowledge everyone who has helped me with this project, our lab members and our collaborators and NIH. And thank you for your attention. If you have any questions, I would be happy to answer.
Thank you, Zina. We have one question so far. Does the electric field generated by neuronal assemblies in native neural networks modify the spatial distribution of the electric field produced by brain stimulation?
For sure, like intrinsic activities, like basically something like effective coupling between neuron assemblies definitely play a role in the spatial distribution of the electric fields. And that would be a very interesting question to answer. But right now, for the simplicity, we ignore that part and assume neurons are just on their own. There we have like one single neuron that is only affected by synaptic inputs from the other neurons rather than external electric fields from neuron assemblies. But for sure in the future, we can look at the networks of neurons connected together and study those topics, but right now that would be a very complicated question to answer with these level of details in our multi-scale modelings.
Thank you. One more question. Could you comment on how you couple your neural models to the electric field models for tMS exactly on a physical level?
Yes. So basically, we used Dr. Peter Cheff's method for easily coupling electric fields to the neuronal level. So what is important to note that neuron package has something called extracellular mechanism, that if you provide the external potentials, like basically electric potentials at the external level at each of the nodes in the neurons, neuron package can automatically simulate what happens at the neuron level under such electric potentials. But it is important to go from the electric fields to such potentials. And basically, we use quasi-potential estimation, which is like getting the integral of the electric fields across all the branches in neuron. For example, let's start from soma and assign a quasi-potential of 0, because in quasi-potential, the exact numbers don't matter, only the shifts, like variance of the quasi-potentials between at different locations matter. It's like electric potential. And let's say we start from soma and we assign 0 to quasi-potentials at soma, and we start integrating over each branch with the realistic electric fields from the SIMNIBS results throughout each branch. And that way we can calculate quasi-potentials at each location for the external environment of each neuron compartment. And after that, by providing these values to the neuron package, we can automatically take care of that and simulate what happens to the neuron. Hope I answered that question.
May I, thank you very much. Actually, may I follow up? Do you also think that is really what's happening on a microscopical level around the neuron or in the neuron?
So that is, in general, it is very important to always validate the neuron modeling, like any kind of modeling. I do not like claim that these models are accurate and have the answer to everything.
Yeah, I'm not so worried about the models, but about the interface. But you model the outside of the neuron. You set potentials outside, but
yes.
Magnetic simulation is a little bit different than electrical simulation. It doesn't care if it's inside or outside, so it uses on both sides. Right? How can we? So, right, if you just copy it over and over from model to model, and everybody does the same, doesn't make it true. So the question is, do you see an option how we can figure out what's really going on there, and how we could maybe even somehow validate it?
Yes, okay, that's a good question. So first of all, I would want to mention that the electric fields that we get at the like large scale head models in the FEM models assume basically we have an isotropic medium in terms of electrical properties in the like, let's say, gray matter. And that doesn't care about what exactly is happening at the neuron level. We just say there is a general isotropic, let's say, conductivity in gray matter. And after we have those large scale electric fields, by using quasi-potentials, we can like measure what happens at the exterior of the neuron compartments. That is what is happening physically. And cable equation is another physical properties that have been like of what happens at the neuron level, and it has been validated many times before. And I like, this is basic physics that if we put these things together, it should, like, physically speaking, give you the accurate results as far as I can tell. But for your answer to your second question, if we can experimentally like validate the results, for example, one thing we can do, and I mentioned this, is you can have in vitro models. And it's like you can use patch clamping to measure activity at the single neuron level, and at the same time using the in vitro models in SIMNIBS, you can do the multi-scale modeling and see what happens at the neuron level and validate that.
Okay, thank you, Stefan, for the question. And let's say the final question from the audience. Can you comment on computational complexity of this approach? How long it takes to run such neuronal model, cellular to subcellular model?
Right. So you know how long it will take for the electric field modeling. Depending on the complexity of the model, it would take around maybe 10 minutes to run a whole head simulation. For the neuron level simulation, it would take much longer, but of course it really depends on how complex your neuron model is. For the models that I showed you, like for the pyramidal cell, they are very sophisticated models and they take a lot of times. For example, for the tACS results that I showed you, let me go. For these results, basically, I simulated what happens to the neuron like for the duration of 4 minutes. And it was taking me maybe like 2 hours to simulate like 4 minutes of simulation.
On a regular PC, right?
In a regular PC, yeah. And but for like some other analysis, we have run everything on like a supercomputer, on the server, and we can run everything in parallel, and that greatly helps with the speed of the processing.